Well, it has some genuinely surprising twists.
That's right, 2DG. You might also see it written as 2-deoxy-D-glucose, maybe D-arabino-2-deoxyhexose. It's essentially a sugar, but slightly tweaked.
And for anyone looking it up, the CAS number is 154,17,6.
Right, and the key thing, the tweak, is tiny, just one specific hydroxyl group swapped out for a hydrogen atom.
Seems like such a simple change, doesn't it? But this is where it gets fascinating.
It really is.
Our mission, really, for this deep dive is to understand why. Why does this small structural change lead to such a huge range of biological effects? We're talking cancer research.
Potential COVID-19 treatments.
All sorts. We've pulled together a load of scientific reviews looking at how it's made, what it does, these applications.
And it is quite striking. You tinker with something fundamental like how cells use sugar, and suddenly you see effects across the board. Cancer, inflammation.
It really underscores how central energy metabolism is, doesn't it?
Absolutely. And understanding 2DG, it's not just about this one molecule. It kind of gives us a window into how energy use is tied up with so many different diseases.
You see how hitting these metabolic weak points could be a real strategy.
Exactly. Exploiting those differences.
Okay, so let's get down to basics. What makes 2DG special? What does this modified sugar actually do inside a cell?
Well, its main job, its core function, is as a competitive inhibitor of glycolysis.
Glycolysis, that's the main pathway cells use to break down glucose for energy, right?
Precisely. Think of glycolysis like the cell's engine, using glucose as fuel. 2DG is like a key that fits the ignition but won't turn.
Right.
It gets in there, competes with the real fuel glucose for the enzyme machinery.
And jams it up.
Exactly.
Ah,
It jams the engine because it can't be fully processed like glucose.
okay. That analogy makes sense. And I remember reading about cancer cells and their weird energy habits, the Warburg effect. They love glycolysis.
That's the one. Cancer cells often rely heavily on glycolysis, even when there's plenty of oxygen around, which is unusual. Normal cells usually switch to a more efficient process in oxygen.
So this reliance makes them vulnerable.
Very much so. Because they're so dependent on that glycolytic engine, something like 2DG that jams it up can hit them particularly hard. It can hinder their growth, potentially selectively.
Selectively, that's the crucial word. We don't want to shut down energy in healthy cells.
Right. And healthy cells usually have more metabolic flexibility. They're like hybrid cars. If the glucose engine is jammed, they can often switch to other fuels, fatty acids, ketone bodies.
Ah, okay. So they have backup options that many cancer cells might lack.
To a degree, yes.
Yeah.
That difference is key to its potential therapeutic window.
It's like targeting the gas guzzlers while the hybrids can keep going. Okay, what else is 2DG involved in? Something about mimicking energy restriction.
Yes, it's sometimes called a potential energy restriction mimetic agent, or ERMA. By blocking glucose use, it tricks the cell into thinking energy is scarce.
Like mimicking calorie restriction.
Kind of, yes. And this can trigger certain protective responses in the cell, things normally associated with low energy states.
Interesting. And I saw a mention of PAMPs, pathogen-associated molecular patterns,
Yeah.
inhibiting those.
Right. PMPs are like danger signals from microbes, bacteria, viruses that alert our immune system.
Red flags.
Exactly, red flags. And 2BG seems to be able to interfere with these signals, suggesting it might modulate the immune response.
Okay. And we should also mention the tracers. You can stick a radioactive label on it.
Yes, precisely. Radioisotope derivatives, like FDG, which uses fluorine-18, are incredibly useful tracers in medicine.
Like in PET scans, to see where glucose is being used up rapidly.
Exactly. Brain activity, finding tumors, inflammation. Very useful.
And then, jumping ahead a bit, its use in India for COVID-19. That definitely caught people's attention.
It certainly did. It got emergency use approval there for moderate to severe cases. The rationale partly circles back to what we said about cancer cells.
High energy demand.
High energy demand, reliance on glycolysis. Virus-infected cells can also ramp up their metabolism. Early reports suggested it might help patients need less supplemental oxygen.
So the same principle targeting high energy needs might apply beyond cancer. Fascinating.
It really connects different disease states through metabolism.
But you mentioned it's not just about jamming the glucose engine. There are other things going on too.
You're absolutely right. It's not quite that simple. Its structure, it doesn't just resemble glucose. It also looks a bit like another sugar, mannose.
Okay. And why does that matter?
Well, that resemblance means it can interfere with another really crucial process called N-linked glycosylation.
Glyco what now?
Glycosylation. Think of it as Adding complex sugar chains, like decorations, onto newly made proteins. These decorations are vital. They help proteins fold correctly, tell them where to go in the cell, how to interact.
So like address labels and instruction manuals made of sugar?
That's a good way to put it.
Okay.
And 2DG, because it resembles mannose, which is part of these decorations, can get incorporated incorrectly or just generally mess up the whole process. It's like using faulty decorations.
Okay, faulty decorations gumming up the works and disrupting protein production or function. That sounds like it could definitely have antiviral effects.
Precisely. Messing with glycosylation is thought to be one way it hinders virus replication. And it doesn't stop there. This also links to its anti-inflammatory effects.
How so?
Many key proteins involved in the inflammatory response, think cytokine receptors on immune cells, they need those correct sugar decorations to work properly. If 2DG interferes with their glycosylation,
It can dampen down the inflammation.
that seems to be the case,
Yeah.
yes. It can help regulate the whole process.
Wow. So you mentioned it being looked at for respiratory infections, even something like genital herpes, HSV.
Yeah, there's research there. For instance, in studies on HSV infections in the eye, using 2DG to limit glucose seemed to reduce the amount of inflammatory damage, the lesions. It appears to affect the inflammatory mediators somehow.
And you mentioned PMPs earlier, the bacterial red flags. It helps with inflammation caused by those too.
It seems so. There were mouse studies where 2DG reduced acute lung inflammation triggered by those PMPs.
So it might be acting quite broadly against inflammation in certain contexts.
It shows that potential. Other studies suggest it can reduce inflammation linked to things like glycogen buildup in immune cells exposed to, say, coal dust.
Okay.
And it's shown promise in animal models for conditions like inflammatory bowel disease, arthritis, even severe systemic inflammation like LPS shock, possibly through that glycosylation inhibition route we talked about.
That's quite a range. And then the really surprising one for me was the brain connection anti-seizure effects, maybe even slowing epilepsy. That seems unexpected for a sugar analog.
It does feel like a different category, doesn't it?
Yeah.
But research suggests it might influence a specific signaling pathway in the brain involving something called netrin G1 and potassium channels, KATP channels.
Okay.
And we know brain metabolism is altered in epilepsy. You get complex changes in energy use. So the idea is that targeting glucose metabolism with 2DG might help stabilize brain activity, potentially reduce seizures or slow the disease progression.
Right. Hitting that metabolic angle again, but in the brain this time. Okay, let's circle back to COVID-19 then. We mentioned the emergency use, the rationale about energy demand. Can we unpack that a bit more?
Sure. The core logic, as we touched upon, is that similarity between rapidly dividing cancer cells and cells heavily infected by a virus like SARS-CoV-2.
Both are energy hogs, basically, and
Yep.
rely on glycolysis.
Pretty much, yes. And lab studies in vitro work showed 2DG could inhibit the replication of several viruses, and that included SARS-CoV-2.
So the idea was cut off the energy, stop the virus building copies of itself.
Exactly. Alt energy production and viral synthesis in those infected cells. Plus, some computational work, docking studies, suggested 2DG might even interact directly with some of the viral proteins themselves.
Interesting. It's really something how this metabolic link connects cancer and viral infection. And you said some research suggests this ramped up glycolysis might actually make COVID-19 worse.
That's a key point. There's evidence suggesting the virus itself can push infected cells, maybe even in organs like the heart, towards more glycolysis, and this metabolic shift could contribute to the severity or complications of the disease.
So 2DG could potentially counteract that harmful metabolic shift.
That's the hypothesis, yes. And people are exploring variations too, like combining 2DG with hyaluronic acid as a delivery strategy for COVID-19. And studies have shown it blocking viral replication in specific cell lines in the lab.
It all sounds very promising, but there have to be downsides, right? Toxicity concerns. It can't be completely harmless if it's jamming up a core process like glycolysis.
No, you're absolutely right to be cautious. It's not without risks. That interference with glycosylation we talked about, that can have broader effects on normal cell function too.
And at higher doses.
At higher doses, yes, toxicity is a concern. Effects like lowered blood pressure, slowed breathing have been noted in some studies, usually at higher concentrations. There's a maximum tolerated dose, often cited around 250 milligrams per kilogram in animal studies, but clinical doses explored are usually lower, maybe up to 63 or even 90 milligram per kilograms per day in some recent trials.
And side effects in humans.
Things like reversible high blood sugar have been seen, which makes sense if you're blocking glucose use. Gastrointestinal issues, even bleeding at higher doses, have also been reported in some cancer trials, so dose is critical.
Finding that sweet spot, enough to work, not enough to cause significant harm.
Exactly. It's always a therapeutic window calculation. Interestingly, some think a ketogenic diet, which forces the body to use fats instead of glucose.
Might make cells more tolerant to glycolysis blockers like 2DG.
Potentially, yes. It's an area of research. We
Oh.
also need to consider resistance.
Cells figuring out a way around it.
It can happen. Some organisms might, for example, start producing more of the enzymes that can handle 2DG or bypass the block.
And before we move off risks and uses, we should definitely re-emphasize its role as a research and diagnostic tool, the PEP scan.
Oh, absolutely. That's perhaps its most established and widespread use. Using radiolabeled 2DG, particularly FDG with fluorine 18 for PET scans, is standard practice.
To see metabolic activity.
Yes, assessing brain function, pinpointing cancer metastases, tracking inflammation. It's an invaluable diagnostic tool, completely separate from its potential therapeutic uses.
Okay, great. So we know what it does, the good and the potentially bad, but how do scientists actually make this stuff? Synthesizing molecules like this isn't always easy.
No, it often isn't. And while there are quite a few ways to synthesize 2DG described in the literature, yeah, many can be a bit fiddly, multi-step, maybe low yield.
What kinds of starting materials do they use?
Often they start with readily available sugars or sugar derivatives. Glucose is a
Yeah.
common one, derived from glucose itself.
And the chemistry involves.
Often things like adding a bromine atom selectively, then replacing that bromine with hydrogen. Chemists use various tricks for that photolysis, which uses light or catalysts like Randy nickel. Some of these routes report pretty decent yields for certain steps, 78%, even 95%.
Classic organic synthesis. Can they start directly from glucose?
Yes, there are methods for that too. One was published that goes from D-glucose in just 3 steps, supposedly with around 48% overall yield, which is pretty efficient for this kind of thing. It avoided needing lots of protective groups, simplifying things.
Okay. Any other starting points?
Sure. Things like breaking down sugar alcohols called tetrols using ozone. You can even start from other sugars like dimanose or ribose.
So quite a few chemical routes. What about using biology's own tools enzymes?
Ah, yes, enzymatic synthesis. It's definitely a more modern and often greener approach. Researchers have used enzymes, like one called aldolase from rabbit muscle, to make 2DG.
With good results.
Reportedly very good, yeah. One study mentioned 95% yield in the final step. There are also enzymes from fungi, like Aspergillus niger, used to make related 2-deoxy compounds. Enzymes can be very specific and work under mild conditions.
So it sounds like making 2DG is getting more efficient, which is probably important if its therapeutic uses take off.
Definitely. Finding economical and scalable synthesis methods is crucial, especially with the renewed interest sparked by things like the COVID-19 research. Reliable supply is key.
Right. Well, this has been incredibly insightful. We've gone from a tiny molecular tweak to cancer, viruses, inflammation, epilepsy. Quite the journey for one molecule.
It really is remarkable.
So if you had to boil it down, what are the main takeaways for everyone listening?
I think the core message is how a seemingly small change, removing one hydroxyl group from glucose, unlocks this huge cascade of biological effects, primarily by messing with energy metabolism.
Hitting those energy-hungry cells.
Right, cancer cells, virus-infected cells. But it's not just energy, it also impacts other key processes like protein glycosylation and inflammation signaling.
It's like one key fitting or jamming several different locks in the cell.
That's a great analogy. And that multifaceted action is what makes it a potentially valuable therapeutic agent for such diverse conditions. Cancer, viruses, inflammation, maybe even epilepsy. Plus, its role as a diagnostic tool remains incredibly important.
Absolutely. So let's leave our listeners with a final thought. We've seen the profound effects of tweaking glucose metabolism with 2DG. It makes you wonder, doesn't it? What other seemingly minor modifications to fundamental molecules, other sugars, amino acids, lipids, might hold similarly unexpected, perhaps even beneficial pharmacological secrets?
That's a fascinating question. It really highlights how much potential might still be hidden in the basic building blocks and processes of life. Lots to think about there.